But now onto the business of the day. I think you're waiting for this a lot more than you're waiting for me. And without further ado, I call to the podium our chair of the day, Colin Honey. And Colin, it's wonderful to have you back here in person at the Sofitel. I call on Colin to take over today's meeting. Thank you.
Thank you, and I'd first like to add my recognition to George, who has recently so sadly died. He was such a beautiful person and such a splendid representative of you people that whenever I met him, his kindness was everywhere to be seen, as it is among the other members of the Rotary Club of Melbourne. And so we celebrate his wonderful life and give thanks for it. Today, we recognise and give thanks for the work of a splendid researcher, Andrew Pask. Professor Pask is a professor, that for a start is pretty impressive, at Melbourne University, which is very impressive indeed, and is recognised throughout the world for his work in genetics and in the work particularly with the Thylacine Integrated Genomic Restoration Research Program. And we can be delighted to hear from him and excited at the prospect. On the other hand, one remembers the cane toad and the those big carp fish in the Murray River, and the importance of the community as a whole understanding the implications of the research and imposing appropriate controls. The field of bioethics, which is mine, began with the Nuremberg trials after the Nazis did such interesting things as freezing people to death who were prisoners, to see how high the members of the Luftwaffe could go without suffering the adverse effects of freezing. It's possible for us in our wishes to do good, to do monstrous evil or to commit mistakes. So there are two sides to this wonderful research that's being done, and we today are being given the opportunity to hear, to learn the facts, to appreciate the great work that's been done, and to formulate appropriate responses to it. And so it is my very great pleasure on behalf of you all to welcome Professor Andrew Pask and invite him to address us.
Thank you very much. All right, well, thanks for having me. That was a terrifying introduction. So hopefully I can convince you all that this is a really good idea and that we have thought a lot, very long and hard about all of the bioethical issues as well. So what I'm going to talk to you about today. So I'm not going to tell you about the science today in detail. Quite frankly, it's quite boring what we have to do to actually bring this back, and very long and arduous. But I'm really going to talk about why we want to do it and why it's so important. I think you heard already that today is marked by the extinction of the thylacine. That's really what marked Threatened Species Day. And the reason that is because it's one of the very few species that we know the final moments, the final date that it existed on this planet. And it's a really tragic tale of human-driven extinction that wiped this species out. And so I'll talk to you about why I think it's so important to bring this particular animal back, and the technologies that we're developing along the way that have enormous implications for ecosystems, for Australian marsupials, that are desperately needed because we are not very good at looking after our fauna in this country, unfortunately. So this is that archival footage, you've probably all seen it before, of that very last Tasmanian tiger. This is the one, Benjamin, that died on this day in 1936 and marks that World Extinction Day or World Threatened Species Day. You can see it's this incredibly unique marsupial species. So although it looks exactly like a wolf, it is a marsupial. It had a pouch just like any other marsupial had. It had, about up to 4 pouch young in its pouch. But it was this incredibly unique body shape and design. And what was really unusual about this animal is that it was an apex predator. So they're those animals that sit right at the top of the food chain and eat smaller animals that live beneath them. If you think about other groups of mammals, so the placental mammals, the other big group of all the other mammals that are around on the planet, there's heaps of examples of apex predators. We have wolves, we have killer whales, we've got lions, tigers. You can think of lots of examples of those large animals that sit at the top of the food chain. In marsupials, this was it. We only had one, and it was the Tasmanian tiger. And what that means is when you lose that animal from the ecosystem, because there are no other animals that fulfill its same role or its same niche in that environment, it really destabilizes that entire ecosystem when you lose a species like this. So they're incredibly important in balancing out an entire ecosystem. And it was a non-natural extinction. This was an animal that was persecuted. It was accused of eating sheep in Tasmania. So the Tasmanian farmers really effectively eradicated this species in a very, very short window of time. In fact, at the time, the Tasmanian government was offering a £1 bounty for every dead thylacine that was brought in. And this was around the late 1800s, early 1900s. And it was far more lucrative at the time in Tasmania to become a thylacine hunter than it was to remain a farmer. So many farmers gave up their farms and actually actively went out just hunting thylacines. And so it's one of the fastest eradications of a species that we know of for this animal. The irony of all of that is that they never ate sheep. They didn't have jaws that are strong enough to bite through sheep bones. Any of you who have been to Tasmania, probably most people here will know there's tons of little marsupials running around. There's no need for them to take on a large, formidable animal if they can eat something small and less chewy that's abundant around in their environment. So it was wrongly persecuted for that. Turns out farmers were actually stealing each other's sheep and trying to blame the thylacine for it. So they were actually killing thylacines, stuffing them, and then taking photographs of them with sheep in their mouth to try and instigate this propaganda to get people really, going out and aggressively extincting this amazing species. So it's a really, really tragic tale of this completely human-driven extinction. So just to dispel a couple of questions I always get asked, and have been already since I've been here, is people always say, but they're still out there. We have sightings of them all the time. So I can assure you they are not still out there. This was a photograph that was taken last year. There was a huge press release. Somebody said, we've unequivocally found, evidence of a thylacine. We've got photo trapping of it. And this is clearly the tail end of a wallaby. I've worked on wallabies for long enough to know that this is not a Tasmanian tiger. So there's a lot of claims that we get all the time. One of the incredible things about Tasmanian tigers is how much they looked like a wolf or a dog. So that means anybody seeing, a dog in the distance on a rainy night in the backwaters of Tasmania, or apparently the Adelaide Hills, they've got lots of wine in the Adelaide Hills that might contribute to the problem. They clearly think they see thylacines all the time. So I get emails, letters from people who are absolutely adamant that they've seen thylacines. And for a long time, I explored this. So I would say, if you can send me some scat samples from that area. So I apologise for the picture of the dog pooping and the bag of poo, but my life became, for about 3 years there, people just sending me Ziploc bags of poo from where they thought they'd seen thylacines. And unfortunately for me and the people working in my lab, 100% of the time, it was either dog poo that they'd sent us or Tasmanian devil poo, which is a carnivore as well, so equally as unpleasant. So we actually were profiling the poo and trying to figure out if it was Tasmanian tiger. It never is. So they are not out there. And in the last 86 years, there's never been any scientific evidence of their existence, unfortunately, at all. And you would think within that time, in the small community of Tasmania, you would have at least found one roadkill, one dead specimen that could argue that. So they are not out there anymore, which means if we want to try and bring this species back and, restore it to its environment, we have to work on bringing that animal back from the dead. So then the other thing people always say is, look, I've seen the movie about this already and it didn't go very well, and that we shouldn't be doing this, right? So first I can tell you all that there is no DNA left in dinosaur specimens, so they've been dead for way too long, so we can't bring back a T-Rex or a Velociraptor and relive our Jurassic Park things. But I think also from the bioethics side of things, we really do need to be very careful about when and where we would want to deploy this technology. We can bring these animals back, but we have to think about which animals we would want to do and what's the reason for doing it. And is there somewhere for that animal to live and thrive once more? We don't want to bring back animals just to simply put them in a museum for us to look at and see. We want them to actually be able to replace that lost part of the ecosystem that was gone when they were hunted to extinction. So you don't have to worry about any T-Rexes or any of those things as yet. The real reason that we want to do this kind of biology is not just for bringing back these incredibly unique species that have been lost, but for the important role that they play then in conserving all the other species in that balanced ecosystem. So for some of these really critical cornerstone species, like the Tasmanian tiger, there's a really good reason for why you would want to restore that. And in developing the technologies that we need to bring the Tasmanian tiger back, we're actually developing a whole heap of new conservation tools that we desperately need in Australia for our marsupials right now. And this was also alluded to by Adrian's introduction before, is that we have an absolutely devastating history of extinctions in this country. So we have the highest rate of mammal extinctions in Australia of any developed country on the planet, which is deplorable, I think, for us as Australians. And we currently have 21% of our land mammals threatened with extinction. That is a huge number. 1 in 5 of all of our mammalian species in Australia are on the brink of extinction right now. And we're predicted to have 1 to 2 extinctions of those animals per decade moving forward. And this has increased the risk of this with global warming and with the adverse weather events that we're all familiar with now, like the horrendous bushfires we had a couple of years ago, that can wipe out a number of marsupial species in one adverse weather event. So this is really something that we have to be very cautious of and think about how we're going to safeguard ourselves against this loss of biodiversity, against these loss of these unique ecosystems that we have in Australia. So one of the main reasons we want to bring back the Tasmanian tiger is because it played this critical role. I think often people struggle to understand what that is. For an animal that eats other smaller marsupials, I think we immediately think, you know, this, if we bring back the Tasmanian tiger, it might wipe out tiger quolls or bilbies or Tasmanian devils because it's going to be eating those animals as its prey. But what these animals do, apex predators, is they typically exist in relatively low numbers in our ecosystem. That's because they eat a lot, they have a lot of energy requirements. But what they do is they really pick off and eat the injured and sick animals within populations of those smaller species that sit beneath them. So I'm sure you're all familiar with the Tasmanian devil, the guy that you can see pictured on the screen here, and they almost went extinct in this last decade because of the devil facial tumour disease. So this is a transmissible tumour that they get from biting each other's faces. It spreads from one infected individual to another, and it nearly led to the complete extinction of the Tasmanian devil. In fact, they would have gone extinct if we hadn't gone over and isolated uninfected animals from that population and then brought them back to the mainland here, where we've raised them in sort of an insurance population setting. And now they're being reintroduced back into Tasmania to re-establish those populations. Now, we can model that if you have a Tasmanian tiger around, that apex predator at the top of the food chain, it would be eating these sick individuals, preventing them from persisting in the population and spreading that disease so widely amongst all of the other animals within that community. And so what that means is, if you have your Tasmanian tiger in place, you actually would have prevented the extinction or near extinction of the Tasmanian devil by its own natural biological control. And that's just one example of how that Tasmanian ecosystem has really gone awry since the loss of the Tasmanian tiger, and why re-establishing some of these species is just so important for conserving all of those other animals. And this is just one, and we know that there are multiple knock-on effects for other marsupial species in Tasmania as well as a result of the loss of the Tasmanian tiger. So how do we do this? Well, science, this was really something that was science fiction. We've all seen Jurassic Park and read this book of Jurassic Park, but this is something that really in the last 10 years has become science fact. So we actually have the technology now to bring animals back. What we need to do this is to be able to sequence all of their DNA. So this is their blueprint or genetic code or set of instructions, if you like, on how to build your extinct animal. One of the great things about the Tasmanian tiger is that it's a recent extinction event, 87 years. And so that means that museum collections are actually full of really well-preserved specimens that we can use to get that DNA from. So we can learn everything about how to build our thylacine. So my lab did that about 4 years ago now. We sequenced the entire blueprint of the thylacine, and then we proposed this sort of grand challenge, if you like, of then using that technology to bring the animal back. We also need a surrogate species, a close living relative that could serve as a surrogate for it, and I'll talk a little bit about that. And then development of genetic engineering technologies and assisted reproductive technologies, things like IVF and cloning, that we really need for marsupials to prevent other species from going extinct. So they're all really important technologies that we need right now. This is the most sciencey slide I have in terms of the way that we go about this. But basically the way that de-extinction works is we still can't create life where there is none. So we can't go to a museum specimen, take some of that preserved tissue and then reactivate it or reanimate it and bring it back to life. We still can't do that. So you have to start off with something living and then engineer your thylacine. So the way you do this is that you find the closest living relative to your extinct animal. You look at its DNA, its set of instructions, you compare that to your Tasmanian tiger, in this case. We figure out everywhere where those codes are different, and then we edit that living cell from that living marsupial to basically engineer your thylacine. So you're turning that living cell from a Dunnart cell in this regard, which is this little guy down here, and we're going to engineer its DNA to now become a Tasmanian tiger, to look like this guy up here. And once we've done that, we can use standard assisted reproductive technologies like cloning to turn that cell then back into a whole living marsupial, which will hopefully give us our thylacine that we can then work on studying in great detail before we would consider releasing such an animal back into the environment. So really important to do that second bit. So in terms of what we've done already, we've already sequenced our thylacine DNA. We already know exactly what that looks like. In fact, we've got 95% of that genome assembled, put back together. So we're in a really good shape, better than any other extinct animal actually for the Tassie tiger, which is really great. We already know what its closest living relative is. I'll talk a little bit about that on the next slide. We've already got a colony established of those animals. They're non-threatened marsupials, so they're nice, easy ones to work on, which is good. And what we're doing at the moment is really working on that DNA editing technology, getting those tools in place that we can engineer that entire genome. And that's what we're collaborating with a big biotech company in the States who have given us $10 million to work on this problem, to try and bring this animal back. So we've had amazing support on this because they can see the demonstrated potential of being able to do this and what that would mean for DNA editing technologies for other species and particularly in conservation as well. And then we're working on ways to rear marsupial cells into whole living marsupials and how to raise a little baby on artificial teat, artificial milk, to actually get it into an independent living animal that we could release into the wild. This is our closest living relative of the Tasmanian tiger that we're working on in my laboratory. This is sort of our laboratory mouse model, if you like, for marsupials. So this is a marsupial. It's called the fat-tailed Dunnart. You can see his little fat tail there that he's got, hence his name. But these are amazing little marsupials, very resilient, very easy to work on. They have tons of babies, all the things that we need in order to have a good model to understand marsupial development. But we're developing a lot of the marsupial conservation technologies using this little guy. This would be the animal that would actually serve as a surrogate for the Tasmanian tiger. So you might be wondering, how on earth do you have a mouse-sized marsupial give birth to a Tasmanian tiger? But that is one of the amazing things about marsupials, right, is that they give birth to tiny young. So this particular group of marsupials are called dasyurids. So that's what the Tasmanian tiger is. That's what the fat-tailed Dunnart is. And they actually have the smallest of any of the marsupial babies. So these are about the size of a grain of rice when they are born. So here's just a movie of a brand newborn here that we've just taken to show you them on the day of birth. And they're not even as big as a basmati grain of rice. We're talking your little arboreal grain of rice here. So these are a really tiny, tiny baby. So what that means is that even this little guy here, the little fat-tailed Dunnart, can easily give birth to a Tasmanian tiger. And then once they've given birth, it's a matter of figuring out the milk composition to rear them from that point onwards. And we're actually pretty good at doing that. We can do that for other sorts of marsupials really easily. So we're thinking it won't take us too long, particularly with the resources that we have in the lab now, thanks to the funding, to figure out the mechanism of how we're going to rear that animal from the point of birth right the way through until it's an independent living animal. So marsupials are fantastic for understanding this. We also spent a lot of time understanding everything we could about Tasmanian tiger biology. We want to make sure if we bring this animal back that we're doing it right, and we do have a Tasmanian tiger. And so we were fortunate enough to be able to travel the world and find every single Tasmanian tiger pouch young that there was, the little babies or joeys from their pouches. And we've been able to 3D scan these animals, which has created an amazing resource for museums now, that you can have these sorts of images where we can understand more about the animal. But also it really helped us understand more about the evolution of this species, how it was such a unique marsupial, the role that it played in the environment, and all of those sorts of things. So it's been an enormously fruitful part of my career, being able to unlock the biology of this really incredible animal. But the reason I'm so passionate about this de-extinction science is because of all of the spin-off benefits we get from developing this technology. So one of the key things that we need in order to make this work is to understand and develop marsupial stem cells. So stem cells are the cells that can give rise to every single tissue in our body. You would have heard about them before because they have enormous medical benefits. You know, there's a lot of talk about how these might be able to be used to replace organs for us and other sorts of things that, you know, that we can have any sort of degenerative disease could be potentially rescued by stem cell biology. But you can also take a stem cell or a skin cell from our bodies, turn it into a stem cell, and then use that stem cell to create a clone of yourself or a whole nother living individual. And this is really important technologies for conservation biology and things that we have not yet developed for marsupials. Nobody's achieved developing a marsupial stem cell. We've done it in my lab now, so that was a breakthrough that we had just this year actually, is being able to take marsupial cells and turn them into that cell that can create a whole new living individual. The reason this is so important is because of these increasing adverse weather events, bushfires in particular. We know that we're really running the risk of losing a lot of our marsupial populations. Things like koalas can't flee a fire like you can see here from the kangaroo. They can run away from a fire front. Koalas just literally get barbecued in the tree, right? They can't go anywhere. They're stuck up the tree and they just die. Same as all of the small marsupials, possums and things. So you really do run the risk of losing whole species, or at least a good amount of species diversity when these fires go through. So that's been recognised in Australia now, and there is grants out there for people to go out and collect tissue and biobank tissue from those vulnerable marsupial populations, particularly ones in bushfire-prone areas, to make sure that we have this frozen zoo, if you like, or repository of tissue from those animals. Until now, though, we've never had the technology that if we do wipe out a species, to go back, take that frozen tissue and then recreate those animals to put them back into that environment. We know the bush will regenerate in 3 to 5 years, and we'd like to be able to think now with the technologies that we've developed in my lab, if you've got frozen tissue from animals that lived in that environment, we'll also be able to regenerate that particular species of marsupial or that particular variation that lived in that region and repopulate those environments after a bushfire to protect against further loss of marsupial species. So it's a really important technology. I think you mentioned before the cane toads and the ecological disaster that cane toads has been with the introduction into Australia. So this is something we're also addressing in my lab at the moment as well. One of our most enigmatic little marsupials, this guy here, which is a northern quoll, is going to go extinct because it eats cane toads and it dies from them. Cane toads were introduced here. They did not evolve in Australia, and they're a toxic toad. And as a result of that, none of our mammals that eat frogs, typically, have been able to evolve resistance to that toxin. But if you go to the countries where the cane toad naturally exists, all of the mammals there have evolved over millions of years, just a couple of little changes in their DNA code, which give them resistance to that cane toad toxin. So we know that we can make a quoll resistant to cane toads by just changing 2 amino acids, 2 tiny little letters in its 3 billion base pair genome. So it's 3 billion letters in an entire genome. And we can change 2 of them to make this guy resistant to the cane toad. So that's something we're working on as a really useful application of our DNA editing technology, is bolstering then the resistance of our marsupials to these species. So then not only can we save the quoll from extinction, because now it can eat cane toads, it's not going to not die when it eats a cane toad, but it can also help control the cane toad populations by using them now as a food source. And there are numerous other not just marsupials, but also reptilian species in Australia that are susceptible and dying from cane toads all the time as well. So we'll be looking at engineering multiple different animals in order to have them be able to survive in the face of the environments that we're changing. So I think, you know, the truth of the world today is that we are changing the globe so quickly and so fast through climate change, through the introduction of pest species, that our animals cannot keep up. And unless we are able to step in and give them a hand to accelerate that process of evolution, to bring back some animals that maybe we wiped out before we had a really good understanding of how important they were for balancing those ecosystems, we run the risk of losing even more biodiversity in Australia. So I think we have to have a much broader and more open view about these sorts of technologies and the place that they're going to play in really protecting against biodiversity loss in the future. So de-extinction science isn't just about bringing back animals that have gone extinct. It's really about developing methods to conserve, preserve, and hopefully also restore loss of biodiversity. And with that, I will say thank you very much for listening and take any questions.
Thank you. And in calling for questions, let me ask the first one. Given that we can recreate things that become extinct, do we have to worry about the koalas being eaten up by the fire? Because we can just go to your lab and get some more made, and there will be consequences of that.
Yeah. It's a really good question, right? And I think that people often raise that. Does it diminish then our appreciation of preserving animals if we think we can just bring anything back? And I think de-extinction, at least, so what we're doing for the thylacine, is an incredibly expensive task. It's going to take me 10 to 15 years to make one. You know, this is a really difficult thing to actually bring a whole species back. And it will never be 100% the animal that went extinct. It'll be 99.9% the animal that went extinct, but never that 100% level. There are little bits of the genome, little bits of that code that we can't ever figure out. So there'll always be suboptimal to preserving what we already have. The same goes for recreating or cloning animals from the lab. It's never going to be as good as preserving those species that we have out there. So it's always going to be preservation first, and these are only ever to be there used as a backup strategy for replacing some of those species. But the truth is we're not very good at protecting the environment. We're not very good at preventing bushfires. And so I think we need to have at least some additional tools in our conservation toolkit to fix problems that we know are going to continue to happen.
Thank you. And the members of this club know that they can only be 99.9% perfect human beings, so they understand the problem. Now we have an opportunity for one or two questions. Wait, please, for the microphone to come and announce your name as you ask the question, and then we will have to call the meeting to a close as soon as we've heard. So make your questions nice and crisp.
Hello, Kushal Singhal from Deakin University, studying environmental science. From your research and models, what do you think are the chances of successfully repopulating a population that can thrive in the current environment, especially with the environmental changes since 1936?
Yeah. So I think, you know, one of the great things about the thylacine and why it's such a good candidate for de-extinction is because the environment that it once thrived in is still there in Tasmania. All the animals that it would need to eat to survive and thrive once more are still in place. So the ecosystems don't evolve that quickly. So in the last 86 years, it's still pretty much intact. So that's one of the great things. You know, we're not bringing back something that's been extinct for 5,000 years. We're bringing back something that was very recently here, and you could pop it straight back into its home environment. We're also sequencing about 100 different animals across museum collections. Again, we're such a unique animal that many, many museum collections have thylacine specimens right across the globe. And from that, we've been able to get a really good snapshot of what its population diversity looked like. So the idea would be that we would engineer that back into these animals, that we're not going to bring back one or two, but we would bring back a small population, and they would have a good amount of the variation required to re-establish and thrive back in that environment. But I would imagine this would be something you would study and manage that reintroduction very carefully over many years. You know, we're not just going to let them go and let them do their own thing. They would be very closely managed, and there'd be breeding programs and things to ensure that they could re-establish that place in the ecosystem.
Thank you.
Andrew, Bernie McIntosh. Is this technology transferable to humans?
Yeah, so I mean, I think you've written about this in the past, but we've been able to clone humans for a long time. It's nothing that's been, you know, sort of new. We actually are much better at doing this in humans and have more of the technologies in place than we have for marsupials. But I think that's where it really comes to an ethical question about what you want to apply this technology to and why. But we're very good at creating human stem cells. We're very good at turning those stem cells into embryos. So it's a lot easier than it is for the thylacine.
Thank you. And we could have a dozen Scott Morrisons. How fortunate could we be? Or is that Scott's Morrison? I forget. Next question. No other questions? And probably that hand action from Bernie tells me that the time has elapsed. And we want to thank the Professor for his wonderful talk and to congratulate him on his energy, his enthusiasm, and his success in this area. Melbourne is a wonderful centre of research and of intellectual development, and I know that this club is an example of that, but it's nice to invite some of these young chaps along and they can tell us what they've been up to. But we've been honoured today to receive this absolutely world-class presentation, and I want to thank the Professor and offer him a bottle of uncloned wine.
Thank you very much.
And a pair of socks made in Brunswick. But there we are. Who'd be dead, I always say, and he's the man to say it to. Now, folks, we want to thank those of you who have come today, and it's wonderful to join with you, and to thank also our past District Governor Peter Frewer and Bernie McIntosh for their involvement, and Ross Scholes Robinson, and in fact the whole jolly show. And I mean, you know, it's terrific. One comes just for the speeches. And next week, our monthly evening meeting over at the Girls Club, our guest speaker will be Renzo Mori, who is a sustainability professor and researcher at RMIT, the Royal Melbourne Institute of Technology, formerly the Working Man's College, established by Mr. Ormond. His topic is United Nations Sustainable Development Goals, known in the trade as the SDGs. I wonder what that stands for. So you will be invited to join us there.